Hi, this is the cardiovascular system. I have a question for you. Go ahead. What is the cardiovascular system? The cardiovascular system includes the heart and the blood vessels. The heart pumps blood and the blood vessels deliver it throughout the body. At the center of the cardiovascular system is the heart. It is a muscle about the size of your fist. It's located behind your rib cage and a little to the left of the middle of your chest. The ribs protect your heart. That is why whenever you fall down or get hit in the chest, your heart is safe. Your heart pumps the blood through the body, through arteries and veins. These are like little roads in your body. The blood carries the good, nice oxygen and nutrients that the body needs and carries away the rotten, dirty carbon dioxide and chemical waste so that the body can get rid of them.
Hey guys, this is Mr. Mahmood, and you are watching Energy Lecture 3, or for my honors students, Honors Energy Lecture 4. In this video, we're going to go into the detail behind the second part of the most important series of reactions for all living things. We've already covered the first part in the form of photosynthesis, which is essential for any food chain and any ecosystem in order to make the chemical energy from light energy. But then once that chemical energy is made, it has to be utilized by all eukaryotic cells, including plants and us, to then turn the chemical energy into our activation energy for our cells, which is ATP. So the second part of this is what we consider cellular respiration, and there's a whole lot more to this than just what you might think of as respiration in terms of breathing. So pay close attention to go through the detail here. We're going to use the same set of notes that you should have received in class. Again, have that in front of you. Work your way through it. I'm going to use the exact same diagram and write a lot of things in. Pay attention, try to follow along, try to do the same things. Feel free to pause and rewind and review any of the sections that you may need to really make sure you understand how the whole big picture works, why everything works, and how it all works together. All right, so let's pay close attention here. Now, a good introduction would be to kind of review photosynthesis and then talk about how it's related to respiration. We've done this in class, so I'm just going to move very quickly here. But the diagram here represents the importance of the two being so closely related. So if we look at this top structure, you notice, for one thing, it's using solar energy coming in. So it's something that requires light. You also see the structure. You see these little discs here, so that represents the thylakoids. All of those different sources of evidence would make me suggest that this is a chloroplast. Letter V is a chloroplast. U definitely represents the light energy, so I have photons of light energy coming down, being absorbed by chlorophyll in the thylakoids, again, to allow for the light-dependent side of photosynthesis to take place. The things needed to start photosynthesis, notice I have arrows going from W to X through the chloroplast here, so this top part is photosynthesis. W would be the reactants for photosynthesis, which again are water, which gets split by the light energy, and the carbon dioxide, which is used in the Calvin cycle. And then the products of the light-dependent reaction, when the water is split, produces oxygen gas. And again, the carbon dioxide in the Calvin cycle combines with the hydrogens from the water to make glucose. Again, all of this should be reviewed. That should be nothing new to you, right? So that top part of the reaction is what we consider photosynthesis. Now the key here is that the next step, in order to take those sugars, whether you made them or not, if you're a plant, you made them yourself. If you're any other kind of a consumer, you had to get those sugars from your diet. One way or the other, the sugars make it to your cells. Once you have those sugars, they then need to be used to make your activation energy for your cells, which is ATP. So this Z down here represents the whole point of the bottom series of reactions in respiration. It's all to make ATP. Now you guys remember, ATP actually stands for adenosine triphosphate, right? 3 phosphates. If I use ATP as the activation energy for a chemical reaction, remember what that means. All that means is that I have broken a phosphate by breaking that bond. If that bond is gone, now what I'm left with is ADP plus my separated phosphate, right? So when I say the whole point of respiration is to make ATP, I don't mean it's to make all of these parts separately and start all over from scratch. All I mean is that I need the energy to recreate the bond that'll reassign and reattach the extra phosphate that was separated out to what was there before to reform ATP. Now, remaking and reforming a bond requires energy. So the energy to make that bond comes from your food. So that glucose, whether it's a plant that made it or a consumer that got it from their diet, is used to serve as the energy to make that bond and reattach phosphate so that they can be used again in the future. So the whole point of respiration is to use glucose molecules to make that bond and reattach and reform ATP. So it all happens down here on the bottom half of this series. It happens in two separate places, which we'll talk about. The first part happens out in the cytoplasm, which is the fluid environment of the cell. But the second part, if oxygen is present, occurs in an organelle known as the mitochondria. So mitochondria is the organelle that I want you to think of specifically when we talk about respiration. When oxygen is present, it's called aerobic respiration. Aerobic means in the presence of oxygen. So if we're dealing with aerobic respiration or oxidative respiration, both of those mean with oxygen, then that occurs in the mitochondria. And this is where the most ATP is actually going to be produced for any one glucose molecule. So you'll see in this process, the sugars are needed to start the reaction. Oxygen gas must be present in order for the aerobic part of respiration to happen. So you'll notice the products of photosynthesis are actually turned around as the reactants for this respiration process. And then as the ATP is produced, the other waste products that you create, the water and the carbon dioxide, turn around and become the reactants for photosynthetic organisms. So a plant, if you think about it, can be pretty self-sustained. It can live pretty independently because it makes the products, it makes its own sugars and oxygen, which are turned around and used to create their own ATP. And then the waste products there are actually, again, returned as reactants for photosynthesis. So basically, as long as you have enough light coming in and you have enough water and carbon dioxide to get things started, you can keep the cycle moving. A plant can live pretty sufficiently on its own. Now, organisms like us, consumers, don't have that luxury, right? We can't make our sugars, so we have to get this oxygen and the carbon dioxide from our diet and from the air we're breathing in. But as long as we're getting enough of these, then we can continue to produce enough ATP. And now these carbon dioxides especially is a problem. We need to make sure we get rid of that carbon dioxide so that it doesn't cause any problems in our own blood. So we have to consciously work to bring in these components and breathe out the waste. But a plant can actually utilize it pretty consistently. Now, a lot of times a plant doesn't use all of its glucose and its oxygen, and that excess is what gets released out for the rest of the ecosystem. But it is able to really kind of work pretty amazingly all on its own. So now that you know the basic idea of how respiration is actually the exact opposite of photosynthesis, we can get into the detail of how it all works together. All right, so let's start by writing the basic chemical reaction for respiration. Now, specifically, I'm going to refer to what we consider aerobic respiration. So the aerobic respiration, again, aerobic means in the presence of oxygen, right? If it's aerobic, that means oxygen must be present, right? So we'll definitely start with that. Oxygen gas has to be present in order for the whole process to happen, and I'll explain why. But oxygen must be present, and the basic form of food that's the most efficient source of energy that we've said over and over again is the most efficient source of energy for your cells because it's what your cells know how to use. That is, hopefully you know, a monosaccharide or sugar. The most common example is the one we deal with on a common basis, or glucose, C6H12O6. So aerobic respiration requires oxygen, requires a sugar like glucose, to ultimately produce two waste products. One of those waste products is that you guys know when you exhale carbon dioxide gas. And you guys remember the indicator that was tested out. Had somebody in class exhale with this phenol red, and you can see that it turned orange. It's a carbon or an acid indicator, so it proved that carbon dioxide is present when you exhale. And also, every time you exhale when it's cold or on a cold surface, you should see condensation, which proves that there's water every time you're exhaling as well. So these waste products are produced. I'll tell you exactly how they're produced. But again, the whole point of going through this respiration process is to actually create your ATP. So in the process, if you have everything needed, and specifically you have oxygen available, one glucose molecule will ultimately create 36 net molecules of ATP. 36 molecules of ATP can be created from just one molecule of glucose. Your cells have become super efficient at this process. So balancing it out, let's not even worry about the ATPs, because technically if you're really wanting to balance it out, on this side you'd have 36 ADPs and 36 phosphates, because really all you did if you used ATP was break that third phosphate off. So you're still having all the other parts and you're just recycling it. So if you really wanted to balance that, you'd put it over here. So let's not even worry about the ATP. But everything else in terms of C's, H's, and O's, you guys remember, balance it the exact same way you balance photosynthesis, because it's just the opposite reaction. You put a 6 in front of everything except for that glucose, and that's your balanced equation. Now we're going to keep this balanced equation the way it is, because mathematically I think it's good to try to make it all make sense in terms of where all the elements are going here, without going into too much detail about the explanation. So basically here again, I told you there's a series of reactions that's going to happen outside of the mitochondria in the fluid environment called the cytoplasm, and then if oxygen is present, then the majority of the work will happen in the mitochondria. So we're going to split everything up into two basic parts: when oxygen is necessary and when it's not. So we're going to start with where it's not necessary, because the first series of reactions doesn't actually require oxygen gas. So I'm going to use the red color to represent anything that's not requiring oxygen, and the blue color to represent anything that is. All right. So again, anything that's not requiring oxygen is not going to occur in the mitochondria. It's going to occur out in the fluid environment of the cell, also known as the cytoplasm. So in the cytoplasm is where the first part of respiration will actually take place. It's a series of reactions that's going to break down glucose, literally split glucose in half, into two halves. So that splitting of glucose has a name, and it should make sense to you. The series of reactions that's going to happen here, it's just another cycle. Remember, there's the Calvin cycle in photosynthesis. There are actually two cycles that we're going to deal with here for respiration. The first one is called glycolysis. Let's break it down. You guys remember what lysis means? It means to separate or to break. So glycolysis literally is the separation of glucose, or the separation maybe of glycogen, which is just a long chain of glucose. One way or the other, you're breaking down glucose molecules and separating them out. So glycolysis is the separation of glucose. It's a series of chemical reactions. But the basic idea here is that you need three things in order to allow glycolysis to happen. Oxygen gas is not one of them. That's why I'm writing it here in red. The three things you do need: you need some form of a sugar, like glucose, as your starting point. And again, this is why your cells always prefer to use sugar first, because it's already in the form it needs. It doesn't have to go through a process to convert this into something else, into sugars, which takes its own energy. So your body always pulls from monosaccharides like sugars first. That's why it's the efficient source of energy. This makes sense now. You understand why your cells always use sugars, because this is exactly the series of reactions that requires sugar as a starting point. So sugar is what you need to start. You also need two things. One of them is energy. Remember, every chemical reaction has an activation energy. Now we're dealing with stuff out in the cytoplasm. There's absolutely no chlorophyll here. There's nothing that can absorb light energy, no photosynthetic components here anymore. Even if this was within a plant, all of this stuff happens outside of the chloroplast. So there's no way that light energy can be used. So there is an activation energy here. There's a cost to get things started, and that cost is in the form of ATP. So you actually do need two molecules of ATP as a starting point in this chemical reaction. So ATP is going to be put into the reaction as the activation energy to get things moving. So besides needing glucose, you also need two molecules of ATP to get things started. And you also are going to need, and I'll explain why later, a carrier molecule again to pick up some hydrogens. Basically, you need some more empty buses. Now, the buses this time, if you remember in photosynthesis, they were NADP, nicotinamide adenine dinucleotide phosphate. Now, you just take out the phosphate. They're called NADs. Now, you're going to need two of them, which basically means you need two empty buses to be going into glycolysis as well. So these are the three things needed to start glycolysis. And again, notice how oxygen gas is not one of them. So this will happen whether or not oxygen is present, as long as these three things are there. So glucose we'll go through a series of chemical reactions. And in those chemical reactions, you're breaking glucose apart to basically be split into two halves. Now, those two halves are known as pyruvic acid. So one glucose molecule at the end of glycolysis will produce two pyruvic acids. Now, if you were to think mathematically about what exactly half of a glucose molecule would be, then you would think it would be a certain chemical formula. But the formula for pyruvic acid is actually C3H5O3. What's wrong with that? My math whizzes, take a look at it. The C's are good, right? I have one glucose molecule that has 6 carbons. It turns into 2 pyruvic acids. Each one has 3. So all of my carbons are represented. My oxygens are represented. 6 oxygens split into 2 that each have 3. That's all 6. That's represented. My hydrogens are my problem. You guys see what's going on here? I start with 12, but at the end I have 2 pyruvic acids. Each of those only has 5. So that means I'm left with 10 hydrogens. That leaves me with 2 hydrogens that have nowhere to go. At least not without this empty bus. So that's why you need this NAD. The NAD picks up those hydrogens and will become a full bus. And each one of those will now be considered NADH, right? Also along the process, if you're splitting glucose in half, again, you're breaking bonds. And every time you break a bond, it releases the energy inside. So this process by itself already is able to create some ATP. So this is sort of, an interesting concept. You have to put ATP in to actually make ATP. It's kind of ironic. You can't make ATP without first having ATP to put it in. But the same thing's true for money. You've heard the phrase, you have to have money to make money. You have to be able to invest to hope that you can get some profit coming back. So you invest 2 ATP molecules in every cycle of glycolysis. But in every cycle, you actually end up making 4 ATP molecules. So here's where the idea of net compared to gross amount comes into play. If I invested 2 ATP molecules in, and in the process I ended up making 4, overall, what do I end up with? My net production is actually 2 ATP. Basic idea here, I made 4, but I already put in 2, so I have to subtract the 2 that I already put in. I started out in a negative range, right? I already had 2 fewer ATP than I wanted, but because I made 4, I got back to 0, I got back to even with the first 2, and then the next 2 were just my pure profit. So that's what I'm referring to in terms of pure profit. 2 net molecules of ATP are made every time glycolysis occurs. So just this process alone, I already have more ATP than I started with, which isn't bad, right? So if you're starting a business, already here in this first phase, you've made your profit, which is great. And then from this point on, you're just making more. So that's fantastic. That's what your cells want. Now at this point, 2 ATP isn't really a lot, considering what's possible. You could make up to 36 ATP as long as everything's working efficiently. So let's assume everything's working efficiently. And specifically when I say that, what I mean is that oxygen is present. If oxygen gas is present in the cell and in this fluid environment, then and only then will this next series of reactions be able to happen in the mitochondria. So that oxygen, I'm already going to go ahead and draw it down here. You have to have oxygens available in order for this next series of reactions to occur. And I'll explain why here in a little bit. But let's first talk about the series of reactions. So I'm going to continue, kind of assume the pyruvic acid continues on here. Nothing's happened to it. It's just kind of moving its way into the mitochondria. So again, this is pyruvic acid, C3H5O3. It's going to move into this structure, which we refer to as the mitochondria. The only time components move into the mitochondria is if oxygen is present. Oxygen must be present in order for this stuff to happen. So the pyruvic acid is going to go through the final series of cycles that you guys need to worry about. This is known as the Krebs cycle, named after a scientist named Hans Krebs. He again kind of walked through all the detail of this process, so they named it after him, the same way the Calvin cycle was named after Melvin Calvin for photosynthesis. In this case, it's kind of like the opposite of Calvin's cycle. The Calvin cycle took carbon dioxide, added hydrogens, and made a big thing like glucose out of it. In this case, we're taking something that's pretty big. It's only half of glucose, that pyruvic acid, and it's going to be broken apart. All right, so think about it the same way you're thinking in terms of elements before. So if we have C's, H's, and O's coming into here, getting broken apart, that breaking apart, first off, every time you break bonds, it releases energy. So this by itself will create 2 more molecules of ATP. because you're breaking the pyruvic acid apart. But pretty much the C's and the O's are going to combine here to make CO2, carbon dioxide gas. And for every 1 molecule of glucose, you end up with 6 molecules of CO2 produced from it. Right? So this is where your CO2 comes from, the carbon dioxide waste that you have to get out of your body that goes through your veins, back to your heart, pumped out to your lungs, and you breathe out. It all happens right here in the Krebs cycle, breaking down the pyruvic acid. The C's and the O's combine to make CO2, and that leaves us with hydrogens. Those hydrogens are going to be passed on by more carriers. Again, you have more things like NAD. You actually have another one called FAD, flavine adenine dinucleotide. All of these things are carrying hydrogens over into another series of reactions. This series of reactions is known as the electron transport chain. I'm going to label it over here. The electron transport chain, for most of my students, is probably a new concept. However, for my honors students, you guys know all about it because this electron transport chain is very similar to the same electron transport chain that's actually present in photosynthesis, particularly at the end of the light-dependent reaction. So you have this electron transport chain that functions very similarly. Now, if this is the first time you're experiencing the idea here, we're going to stay very general. Basic concept here is the hydrogens that get sent over technically aren't the full hydrogen. The process of the break actually is pretty aggressive and a pretty strong break. So remember, a hydrogen has 1 proton and 1 electron. So when they're broken, the hydrogen ion is separated and the electron is separated too. So you're left with something that just has a proton, which represents a hydrogen ion. So all of these hydrogen components that were part of the pyruvic acids are now hydrogen ions, positively charged. So they're all building up here in the middle. And then you also have the negatively charged electrons. Those negatively charged electrons are going to be passed from one protein to another. That's what we consider the electron transport chain, the transporting those negatively charged electrons. So you have all of these positively charged hydrogens building up in the middle and these negatively charged electrons building up along a membrane. You guys remember how I told you the mitochondria and chloroplast all have kind of a membrane within a membrane? For a chloroplast, I was talking about the thylakoids. For a mitochondria, I'm talking about this inner membrane. The inner membrane is actually called cristae. Don't worry too much about this, but cristae is the name of that inner membrane. So basically, you have a lot of hydrogen ions on one side of the membrane, a lot of negatively charged electrons developing on the other. And that change of charge actually develops a current, a movement of electricity. And naturally with a current like this, and you'll learn more about this when you get into physics, positively charged things want to move towards the negative. So in this case, hydrogen ions want to pass through into the negative and eventually pass their way out. That movement creates energy. So for every time a hydrogen ion fires its way through the electron transport chain, you create ATP. And in fact, this process creates the most ATP out of any of this reaction. You end up with 32 molecules of ATP just in this step. So this step alone creates 32 ATP molecules, which is amazing considering all the other ones pretty much just making 2. So 32 ATP molecules get made here. And you end up with a whole lot of hydrogen ions out here on the out here on the cytoplasm. And you also end up with a whole lot of these negatively charged electrons. It's a chain ends. So the electrons are getting passed from protein to protein until they're eventually dropped off out in the fluid environment. Now this is where it becomes so important to have oxygen. Now listen very carefully here because this can get confusing. Hydrogen ions determine the pH of any given solution. All right? pH value is really just a value of how many hydrogen ions there are in any given chemical or any given environment. If there are a lot of hydrogen ions, it actually drops the pH. It's kind of opposite, and there's reasons for that that we don't need to go into. It's basically a negative logarithmic scale for all of my math whizzes out there. But pH value is determined by the amount of hydrogen ions. The more hydrogen ions there are, the lower the pH becomes. So if I were to allow everything here in the mitochondria to happen, knowing that there's no oxygen present, then I would allow a lot of these hydrogen ions to build up outside in the cytoplasm. And that buildup of hydrogen ions would actually drop the pH for the whole cell. And if a cell's pH drops, you guys should know this about enzymes, they're very specific. And if anything changes in terms of its environment, the enzyme no longer works well. So if I lower the pH and make the pH too acidic, then the enzymes don't work well, chemical reactions can't happen, and the whole cell can die. So the only way that the mitochondria will allow any of this stuff to work its way through, the only way that it'll allow pyruvic acid to make it in and start the Krebs cycle and go through into the electron transport chain, is if it knows that there is oxygen present for the very end here. Because if there is oxygen present, the hydrogens can combine with the negatively charged electrons and with the oxygens to create the final product of this whole series of reactions, which is something that's harmless in the cell. Water molecules. Your cell has no problem with water, right? But it has serious problems with letting these hydrogen ions build up. So if these hydrogen ions were to stay without oxygen, then the cell would die. So it will not allow any of this to happen if there's no oxygen. So if oxygen is not present, none of this series of reactions will happen. It stops at this point, and instead it's going to jump into this series of reactions we'll talk about here in a minute. But if oxygen is present, it'll allow it, because that oxygen will combine at the very end to take something that's very harmful, it could be too acidic, and turn it into something that's very calm and very tame, which is water. So again, this is where your waste products come from, your carbon dioxide and your water molecules. Again, for every 1 glucose, you make 6 water molecules from it. So this is the basic concept of aerobic respiration. You need glucose and you need oxygen in order to create carbon dioxide gas as a waste and water as a waste, and an overall net production of 36 ATP. Remember, you had 2 net ATP from glycolysis, 2 ATP from the Krebs cycle, and then 32 ATP coming from the electron transport chain. All of those combined gives you your 36 net ATP molecules. So that's the efficient process of aerobic respiration. That's if oxygen is present, you can make 36. Now if oxygen is not present, your cells are kind of up against a wall here. They're in a situation where they need energy, but they know they can't go through the mitochondria and make everything they need there. So they kind of compromise. They say, all right, well, if I can't make all 36 ATP, I'll at least do everything I can to get these 2. In order to get these 2, I need to have the 3 things needed for glycolysis to happen. And just a reminder, your 3 things needed are glucose, a little ATP, and then an empty bus to pick up those hydrogens. If you don't have all 3, glycolysis won't even happen. So let's not even worry about the absence of oxygen, not even worry about the mitochondria. If I don't have these 3, I can't even make my 2 ATP, which is better than nothing, right? So usually glucose isn't the problem. Remember, your cells do a pretty good job. Your body does a pretty good job of storing away glucose for future needs. If you eat too much, insulin pulls that glucose into your liver and helps you store it for future use. That way, when you need it, glucagon releases that sugar and sends it back to your cells. So glucose usually isn't something you're going to run out of as long as you're eating a fairly consistent diet and you're getting enough sugars in throughout your lifetime. You're always going to have a reserve. That's usually not the problem. It's also usually not ATP, because your cells, again, are smart. They're not going to let your cells run to the point where you don't even have 2 extra ATP lying around to start making any energy at all. It'll always keep yourself with a little bit of a cushion. The big problem is going to come in the NAD. If I don't have that empty NAD, then I'm going to have the same problem that I had over here. I'm going to have hydrogens that end up out here by themselves, and again could cause an increase in the hydrogen ion concentration, which ultimately would be a decrease in the pH, making the cell too acidic, again killing the cell. So glycolysis won't happen if I don't have the ability of neutralizing those hydrogens with empty NADs. So the series of reactions that's going to happen here will be just trying to turn pyruvic acids into something that has the ability to carry some more hydrogens that can be picked up and or pulled from the NAD. All right, so there's a series of reactions here, and I'm going to go back to red because, again, this is in the absence of oxygen. There's a series of reactions here that are considered anaerobic. Remember, aerobic is in the presence of oxygen, so anaerobic is without oxygen. So if there's no oxygen present, then we're going to get into this stuff down here in the box, which is still in the cytoplasm. There's no actual new organelle here. I'm just kind of separating aerobic from anaerobic. So the anaerobic part is going to be here at the bottom. It's also known as fermentation. So any kind of anaerobic respiration is also referred to as fermentation. All right, there are 2 categories of fermentation. There's lactic acid fermentation and there's alcohol fermentation. Now, all organisms have the ability of going through one of these two types of fermentation, right? You don't get to choose. Based on the organism, you go through one or the other. If it's lactic acid fermentation, there are a lot of organisms that go through it, types of bacteria, things like that. But we as humans go through this as well. And particularly, we relate to human muscle cells probably the best. Most of us can relate to this, so we'll talk about that in relation to human muscles. Alcohol fermentation is specific to some other categories of organisms. The most common one we use is a specific unicellular fungi referred to as yeast. So yeast go through alcohol fermentation. So let's talk about each one. Now I'm going to kind of separate these out so there's no confusion here. All right, now both of these have the same basic goal. They start with the same components. The reactants are the same. You have your 2 pyruvic acids. Remember that we're made in glycolysis. Same thing here, 2 pyruvic acids, C3H5O3. We'll just label that pyruvic acids. You have a problem, which is the full buses. You have NADHs, but you don't have NADs. And you need to basically turn NADH into NAD. So you have the problem, which are 2 NADs that are the full buses that include the H's. So both of them are starting the same way. Now, what you're wanting to have happen is to turn that pyruvic acid into something that can carry the hydrogen. So I can just basically transfer these 2 hydrogens to this bigger molecule and end up with the free bus again. So in terms of lactic acid fermentation, it's pretty straightforward. The pyruvic acid is turned into lactic acid. Now lactic acid is exactly what you would think of in terms of half of a glucose molecule. So it basically is exactly half of a glucose, C3H6O3. So you end up with 2 lactic acids here. And in doing that, you've freed up your bus. So you have 2 NADs ready to go back into glycolysis and at least let that process happen so you can at least continue to make 2 ATP molecules until you get that oxygen again. Now, your cells usually do a good job of keeping oxygen in high demand and high supply as well. The only times you really are in a position where you run out of oxygen in your cells is when you really exert a certain muscle or a certain group of muscles in your body. So that's when we're talking about heavy workouts. If you're involved in some kind of a heavy workout, you're forcing your cells to use up that oxygen quickly to try to make all 36 ATP as much as they can because you're constantly using it for the activity. And eventually you may run out of that oxygen. And if you run out of that oxygen, if you weren't able to do this, your body would just completely shut down. But instead, it understands that it's that survival instinct in you that you may still need to continue to make as much ATP as you can even though you're out of oxygen. So what it'll do is go through lactic acid fermentation to create at least 2 ATP molecules. You at least want to open the NADs so that you can keep glycolysis going. And remember, every time you go through glycolysis, you make 2 net ATP. So your body will do whatever it can to at least get that 2 ATP production as much as possible until you stop the activity, you again get your breathing rate back to normal, the oxygen is able to make it back into your bloodstream and back to your cells, replenish the amount of oxygen, and then that lactic acid can kind of work backwards, back into pyruvic, and then go into the mitochondria and work everything out the same way. Now that lactic acid buildup in your muscles is what makes you feel sore. So the reason you feel sore after a heavy workout or after a lot of vigorous activity is because for a period of time your cells were out of oxygen. You used it up. So you were forced to go through this fermentation process, and depending on how much lactic acid you built up, it causes swelling in the muscle fibers, and that pressure and that swelling pushed up against your nerves responds as a feeling or sensation of pain. So you feel that pain because you forced your body to work in overdrive for a while. Until your body can get back to normal, you'll still feel that soreness. So that's lactic acid fermentation, just to do whatever you can to get a little bit of ATP through glycolysis. Now, yeast have the same basic goal in mind, but they don't quite go through the same series of reactions. Now, instead of creating something that's such a clean-cut separation like this, they make a product that's known as alcohol. This is why it's called alcohol fermentation. They make ethyl alcohol, or ethanol. Now, the chemical formula for this is not as big as you would think. It's actually C2H6O. This is what we consider ethyl alcohol or ethanol. I'll just go ahead and write out ethanol here. So all alcohols are made this way, and a lot of food components use this process to make the food as well. And I'll explain a lot of this in class, so just pay attention for that. But because this isn't right, my mathematic whizzes out there, see there's a lot of elements missing here. In order for this to balance out, there's a second product besides the NAD that's made here, and it's a product that you're pretty familiar with, carbon dioxide gas. So the same waste product that we talk about happening in the Krebs cycle is produced here as well, because the ethanol or the ethyl alcohol isn't big enough to really take up all the elements. It is big enough in the hydrogen part. Remember, it's all about the hydrogens. It has more hydrogen availability, which means it will take those hydrogens from NADH, and again, the whole goal is reached. We now have 2 NADs that can go back to glycolysis and keep the process going. But because ethanol is smaller, there's a second product in alcohol fermentation in terms of carbon dioxide gas. So make sure you're comfortable with that. So either type of fermentation is just a way to turn pyruvic acid into something that has a little more space for hydrogens so that the NADHs can turn back into NADs so that you can have the 3 things necessary to at least let glycolysis happen. If you have oxygen, then you don't have to worry about converting pyruvic acid to anything. It can quickly just move into the rest of the components of respiration with the Krebs cycle and the electron transport chain and make all of the ATP molecules it can. Remember, the whole key is to keep yourself from getting too acidic. You don't want hydrogen ions sitting around in the cytoplasm. You always have to make sure they're neutralized somehow. And that happens both in the glycolysis process with NAD neutralizing it, and it happens most importantly at the very end of the electron transport chain with that oxygen gas neutralizing the hydrogen ions to turn into water. That's why oxygen is so essential to let the rest of respiration take place. All right, guys, there's a lot to take in there. I know that was a lot to write down. Hopefully you took your time, you paused, rewinded, reviewed any sections that you could to make sure this made a whole lot of sense to you. Pay close attention in class, and don't be afraid to ask questions. Don't forget, all of this stuff is very, very important for this unit for the rest of the year. So, hope you enjoyed it. Take care, guys.